Good afternoon, everybody, and it's really lovely to be able to tell you a little bit more about what we've been doing in the Blewitt Lab with developing new targeted therapies, or attempting to develop new targeted therapies for Prader-Willi syndrome for this family day. So today I'd like to tell you about two approaches that we're taking to activate the silent genes in PWS, and explain a little bit about why we're taking two approaches rather than just one. So, as you all know, the cause of Prader-Willi syndrome lies in a suite of genes that are found on chromosome 15. So, like with all genes, you inherit one copy of these genes from the father and one from the mother. But in this case, for Prader-Willi syndrome, these genes are actually only active or being used from the copy you inherit from your father. And on the other copy, they are silent. The copy from the mother, they are silent. And in patients with Prader-Willi syndrome, these active or meant to be active genes are absent, meaning that they only have the silent copy from the mother. I'd like to take a moment to explain a little bit more at what I mean about active and silent genes. So here, what I've got on the left is a picture of what happens normally in a cell. You have the DNA, so the genes that are found in the nucleus, and these genes are transcribed into a copy called an RNA molecule, in this case a messenger RNA molecule. And that's because you need to keep your DNA in its double-stranded form in the nucleus of the cell, because this is really the most critical information that the cell has, and it needs to be kept as safe as possible. But also that genetic information is the information required to create all of the proteins and the molecules that are required in the cell for it to function. So the way the cell gets around this is that it makes this messenger molecule, this RNA molecule, which is an identical copy of the DNA of the gene, but only has one single strand that's shown here rather than two. So it's less stable. It's really just a transient product. And this is then exported out into the cytoplasm of the cell, where it can be made into the amino acid chain, which then folds into the protein as a result. So active genes, those genes that are being used in the cell, are the ones that are being transcribed. And then they can go on and be made into their protein product. Silent genes, on the other hand, are not undergoing this transcription stage in the first place. And that's really the difference between the two. They're not going on to make their products that are used in the cell. So the product of an active gene can be the protein, and this is often the case, but they can also sometimes be this RNA in between. And this is indeed what's true for some genes that are important in Prader-Willi syndrome, is that they don't make a protein in the end. They stop at this RNA form, and it's the RNA that does the work. So if we come back to Prader-Willi syndrome and remember that we don't have the active genes, a suite of active genes on chromosome 15, and we only have the silent ones, that means they still have the DNA that's there. They still have the genetic information. Patients all have that genetic information, but these genes are not being transcribed. And one of the reasons is that this protein that we work on, called SMUCHD1 or SMCHD1, SMUCHD1 we call it affectionately, and we know that SMUCHD1 contributes to these genes not being transcribed, these genes being silent. So therefore, one potential treatment for Prader-Willi syndrome is to remove SMUCHD1, and that will then hopefully switch these genes back on again, and therefore we can use that genetic information that all the patients have. So the question then becomes, how do we do that? How do we actually remove SMUCHD1 function? And there are really two ways to do this. And what I've told you about before, and I'll give you an update on today, is developing chemicals that can inhibit SMUCHD1's enzymatic activity. So just as a reminder, chemicals that inhibit an enzyme are really the most common form of drugs that we use, and they've been around for a very long time. So enzymes in the cell are really the ones that do the work. They take something and convert it into something else. So in the case of SMUCHD1, it burns the energy of the cell called ATP, and that's really just like the cash in the cell, and it burns it and is able to do some work on the DNA actually. So what we need to do to develop chemicals is these chemicals need to somehow break that enzymatic activity. And so we're trying to, and that's really what most drugs do. They break this enzymatic activity. And penicillin and aspirin are really good examples of that. But the second avenue that we've been investing in also in recent years, in the last year or so, has been a second type of therapy called antisense oligonucleotides. And these can, instead of actually just breaking the protein that's there, they can instead have the capacity to deplete the protein entirely. And therefore, you just don't have SMUC-D1 around to do its job at silencing these Prader-Willi genes. And so the reason that we've started on this second avenue that I'll tell you a little bit more about today is because it's a very new type of therapeutic, and it's being used successfully for other developmental disorders. And there are a few key advantages. And I'm going to come back to those at the end of the talk. But just suffice to say at the moment, the reason that we want to have 2 strands is because it's always better to have 2 chances than 1. And given that we know when in drug development that many drugs don't, despite lots and lots of investment and time going into developing them, they don't all make it into the clinic. But if you have 2 chances, you've got a better chance of getting there in the end. And so we think this is a really good plan. So when we come back to that first type of approach, I'll just move my image. We know that to be able to find chemicals that inhibit SMUC-D1's enzymatic activity, we really need to search through many hundreds of thousands of compounds to find some that do that. And then we work to improve them. So we've done this before, and from our prior work, we have one series of chemicals. They're all related chemicals but one series is not enough. So just like I said, having one avenue, so chemicals or antisense oligonucleotides, ideally you have more than one. Similarly, even within each of these pathways, it's much better to have a second series or even a third. So what we started to do a year and a half ago or so was to perform a high throughput chemical screen do a new high throughput chemical screen. And so this involved us screening through more than 284,000 compounds or chemicals. And this was 227 plates that each had 1,536 wells in them. So here is an image of one of those plates and somebody's hand behind it. So you can see how tiny each of those assays are. So from that, we found around about 1% hits. And last time I gave an update in 2022, this is the number of hits that we had, and we're currently working further on them. So this is actually quite a high number of hits. And so we actually went against what we would normally do. And we brought in our medicinal chemists at this point. And the medicinal chemists went through and with their expertise, were able to look at each of these types of hits and whittle down the list to a much more likely size or number of hits, around 1,000 potential hits. And with this number, that was a much more feasible number to be able to go through and validate in our subsequent assays. And so the first thing we did was, with all 1,000 compounds, we tested them not at 1 dose of drug, or 1 dose of chemical, but now at 10 different doses. And so you can see, first of all, whether these chemicals actually behave as you would expect them to when you have higher or lower amounts of chemical. We then took the 10 best of these and put them into a separate assay entirely to see if they would still read out, if you like, still look like a hit. And so all of these assays, what they're trying to do is send this large number of compounds through a pipeline to try and whittle down to the one that's most likely to be able to do the job of inhibiting SMUC-D1 that we feel most confident about. And so by going through this process, we came out with one new lead molecule that we chose to take forward, and we've started to work on further at the beginning of 2023. So the stage that we're at now is to perform medicinal chemistry. So I mentioned we'd already been doing a little bit of medicinal chemistry on the very large number of compounds, and that was in silico in the computer. But now what we've been doing is trying to work on that single lead molecule and try and increase how potent it is, so how well it can inhibit SMUC-D1. And we're then next going to work on how selective it can be. So what medicinal chemistry really means is intelligent drug design, I guess. And so if you think of these chemicals that we want to develop into drugs later on as being a key, they need to fit precisely into their lock, and their lock in this case being SMUC-D1. They need to fit precisely into SMUC-D1 as the lock, but no other lock in the cell. So nothing else that looks a bit like the same lock, because you don't want this to open anybody else's lock, if you like. And so what we're doing is we start out with this single hit that we have that looks a bit like this tablet here. And so it can be jammed into the enzyme, into the lock here. but it's really being jammed in. It's not going to work very well. And so what we have to do is make minor modifications serially. So you make one small modification, see if it makes it better, make another small modification, see if that makes it better or worse, another. And so that's what we've been doing, and so far with several rounds this year. And what we want to do is to create something that fits perfectly and therefore is really potent, and then next work on making it selective. So what we've currently done is tested 40 brand new chemicals that were synthesized by our medicinal chemists, and we've increased the potency excitingly 1 to 200-fold. So this is really amazing, and actually we're surprised that this has worked so well in the time frame so far. So next, what we'll do is continue to increase potency and also work on increasing selectivity. So making sure it only fits in this green enzyme and not a blue one, a pink one, or a red one. So with potent and selective chemicals, so once we've got to the end of this stage, and we will to go alongside our prior series, we can then take these compounds and they could be developed, these chemicals, and they could be developed into drugs. Now, this tends to happen in a commercial environment rather than in a research environment because of the scale that's required. So the next thing that I'd like to tell you about are explain how antisense oligonucleotides work, because this is something that I haven't spoken about before. So if we come back to this picture I showed you earlier about the DNA being transcribed into RNA and then made into protein, then where does these oligonucleotides really are DNA or RNA. They're acting at this level, and that means that they can be designed to be really perfect for exactly the RNA or the gene that we're working on. So in this case, what we have is an antisense oligonucleotide, and it binds to the RNA molecule, and it does so because it is made up of the same bases or matching bases that are found in the RNA of SMUCD1. And what an antisense oligonucleotide does, by creating this little bit of double-stranded RNA, which is, and remember, RNA is normally single-stranded, double-stranded RNA is then the target for another enzyme in the cell, this little Pac-Man. And this other enzyme in the cell then munches up all of that RNA and degrades it. So this is a really beneficial process, because what you can then imagine happens if you munch up the RNA, then there's nothing to be exported and made into the amino acid chain and folded into protein. So it's a way to very specifically, because you're using this antisense oligonucleotide that is directed only to this specific nucleic acid sequence in the RNA, very specifically target the production of the SMUCD1 protein, or in other cases, other genes. So what we're doing right now is we have a SMUCD1 antisense oligonucleotide, SMUCD1 targeting antisense oligonucleotide, that has been developed by Merlin Thomas at Monash, a new collaborator that's really important for this work. And we're now trying this to test it for effectiveness in neural-derived cells from Prader-Willi syndrome patients that we've derived in a dish. And so at this stage, we want to make sure that it could be effective. And if it can be effective, then it can be a second strand of potential therapy. So why might we actually want to have these two approaches? I mentioned earlier just the general kind of idea that it's always good to have more than one iron in the fire, if you like. But I think that there are different advantages for each of these two systems. And so in the end, we'll need to choose one to go forward with. But for the moment, it's a really good idea to try and have both for a little while. So I mentioned earlier that the chemicals that we have that could be developed into drugs, this is really the most standard way of developing a drug, and it's the one that's been around for the longest. And so it's a very well-trodden path, and there's plenty of experience worldwide with this approach. It's a really key advantage. But I think the other really important advantage is that if we're able to develop chemicals later into drugs, then these drugs may be able to be delivered in a relatively simple way. So either by oral dosing, so taking a tablet or a syrup or something, or maybe by injection, but a standard injection, an intravenous injection, or maybe a subcutaneous injection, maybe other routes, but a relatively standard way of delivering medicine. But the disadvantages are that it can be very slow to develop these drugs, the chemicals into drugs, and a lot of it, sometimes you reach hurdles that can't be overcome. one of the hurdles in particular is that we have a very strong blood-brain barrier in our body. And this means that this protects our brain from infection and from all sorts of things that we don't want to be able to get into our brain, because, of course, we must retain our brain's function to live. And so, in fact, infections in the brain, like meningitis, are incredibly rare. So this is a blood-brain barrier is really important for our health. But in our case, what we need is for this medicine, the drug needs to get into the brain for Prader-Willi syndrome treatment. So that poses a challenge, and something that much of the world is working on, actually. On the flip side, for antisense oligonucleotides, they're really not an old-fashioned approach and something that we know a lot about. They're very new in the scheme of therapies, but they are now being used to treat neurodevelopmental disorders, such as they are being trialed for Angelman syndrome. We don't know if it works. That's a related disorder, but also for other developmental disorders like spinal muscular atrophy, Duchenne's muscular dystrophy. So they are being rapidly taken up in other developmental disorders. They entirely avoid this blood-brain barrier issue because at the moment their route of administration is via delivery every 3 months or so directly into the spinal column. So that's obviously quite an invasive delivery method and requires, at the moment, general anesthesia every 3 months. So this is a challenge for that delivery, but it gets around one of the major hurdles. Because of the way that they're designed, because they're dependent on exactly the sequence in the base sequence in the gene that you're trying to target, the development time can be shorter than it can be for chemicals. So that's another advantage. But I think the important thing to remember is that there are still major toxicity issues. We don't get around all of the hurdles, and just with only the defect being that you have these invasive treatment regimes. So there are still major toxicity issues that need to be considered with antisense oligonucleotides. It's not as stable. And so at the moment, we feel that it's a really good idea to try and keep working on both for a time until we work out which is the best path forward. And I think having these two irons in the fire, if you like, is going to be a good plan, and hopefully will lead to faster benefit for Prader-Willi syndrome patients. So what are our current goals then? So just to summarize what we're really trying to do, what we're aiming to do is to have ready to be able to partner either with a spin-out company or with a pharmaceutical company enough data to suggest that it is a good plan to target SMUG-D1 and that it might be effective for Prader-Willi syndrome patients. So what this needs is not only these chemicals that I was telling you about, or indeed the antisense oligonucleotides targeting SMUG-D1. That's absolutely critical, but we also need to have data in neurons that we develop in a dish from Prader-Willi syndrome patients. And as I mentioned, we're doing some work on this already. And also data in vivo in an animal model. We're also working on this at the moment this year and into the future to show that actually depleting or removing SMUG-D1 could actually be effective in the context of Prader-Willi syndrome, so that then we could apply exactly whatever drugs we end up wanting to work on in each of these two scenarios, and show that not only can we switch genes back on again, but also that we might be able to have a benefit for the symptoms that are observed in patients. So finally, it remains for me to thank everybody that's been involved. I've given you some very general information, but it's been performed by a very large number of people and also been funded by a very large number of people. So thank you to all of the people in the Blewitt Lab, the Murphy Lab, and the High Throughput Screening Lab at WeHi who have contributed, along with the medicinal chemists who have been absolutely critical, Mel and Thomas at Monash, who I mentioned, and of course all of the funders, including Prader-Willi Research Foundation Australia, but most importantly, the National Drug Discovery